Hello everyone, my name is Tom and I'm a PhD student at the University of East Anglia. Today I'm going to be talking about the causes and consequences of telomere lengthening in a wild vertebrate population, and these are results that are part of a paper that has recently been accepted in the Journal of Molecular Ecology, but they're also available now online as a preprint version. Those who are unfamiliar with telomeres, what they are non-coding repetitive sequences of DNA at the end of chromosomes. They shorten with each round of cellular division due to the end replication problem, and they also shorten when exposed to sources of cellular stress, such as reactive oxygen species. And this shortening generally goes unrepaired due to a downregulation of repair mechanisms in the somatic tissues of adults. These properties of telomeres result in three broadly consistent features of telomere dynamics that we observe at population level. Firstly, telomeres shorten with age. Secondly, life history stress, such as early life adversity, reproductive efforts, disease, these all accelerate the rate of telomere shortening during an individual's lifetime. And thirdly, short telomeres and/or faster shortening is associated with reduced future fitness. Therefore, telomere length and attrition are thought to reflect the costs of past life history on future fitness. In fact, telomeres have been likened to a one-way ratchet model, in that they inevitably get shorter, although the rate of that shortening can vary. Therefore, if you were to measure telomere length at a given time point, it should reflect the stresses that an individual has experienced up until that point, and is therefore a useful metric by which we can compare the rate of aging between individuals. However, we now know that telomere dynamics are far more complex within individuals. For example, here are 4 studies that have assessed telomere length longitudinally, and they each show that telomere length fluctuates widely within the same individual's lifetime. We also see a high incidence of telomere lengthening. That initial observations of telomere lengthening were always attributed to high measurement error relative to the rate of telomere shortening. However, we now know that in many of these systems, the extent of telomere lengthening is greater than we would expect from measurement error alone, which suggests that there are biological mechanisms behind this. But although the circumstances under which telomere shortening occur are well known, the same can't be said for telomere lengthening. We predicted that telomere lengthening would be associated with less stressful environments and life history. This is because telomere shortening is an outcome of more stressful environments, but also because mechanisms of telomere repair, such as the telomerase enzyme, are expected to be costly, and therefore individuals may only invest in such mechanisms under more beneficial environments. Secondly, we predicted that telomere lengthening would be associated with higher survival. This is because of the possible associations with beneficial environments and life history, but it could also be that there are direct health benefits to telomere repair mechanisms. For example, telomerase has rejuvenating effects on the health and aging of lab mice. To test these predictions, we used the extensive longitudinal dataset of the Seychelles warbler on Kuzan Island. This is a small brown insectivorous bird that's endemic to the Seychelles, and this population is entirely closed on this small island, so there's no emigration or immigration to and from the island. So therefore we have very accurate estimates of survival and reproduction for each individual. We also have several measurements of telomere length per individual, as these individuals are recaptured throughout their lifetime. Previous research has shown that telomere length has an overall decline with age, but also that there is substantial fluctuations in telomere length within individuals and a high degree of telomere lengthening, which is greater than what we would expect just from measurement error alone. What we did was calculate the change in telomere length between repeated samples from the same individual. These sample repeats were at least 1 year apart. And during that time between the samples, we calculated how many offspring that individual had, whether those offspring were assisted by the presence of nest helpers, this species being a cooperative breeder. We also determined whether the individual was infected with malaria at the first telomere length measurement, this particular strain of malaria being the only known pathogen to affect the population. And we also calculated the mean food abundance available to that individual. This is because individuals on the islands are confined to territories which vary widely in the availability of their insect prey. We also took into account the PCR plate on which the samples were run. And then we determined whether the change in telomere length predicted the post-sampling lifespan of that individual. What we found was that there were significant associations between the change in telomere length and environmental and life history factors, but only in females. So firstly, we found that females which were initially infected with malaria had shortened telomeres relative to those who weren't infected with malaria. And this mirrors a very similar relationship that's been observed in the closely related greater reed warbler. Secondly, we found that females which had nest helpers exhibited telomere lengthening. And this is particularly interesting in the Seychelles warbler because we've previously shown that females which have nest helpers also have higher survival and longer lifespans. Thirdly, we found an interacting effect of food availability and offspring production, so that females with more offspring had shortened telomeres when the food availability was low, but that they had lengthened telomeres when the food availability was high. So, yes, we found that telomere lengthening is associated with less stressful environments and life history, but interestingly, only in females, not males. One likely explanation for this is that in the Seychelles warbler, reproductive effort is biased towards females, since it's females that produce the eggs. Only the females incubate the eggs, and they do the majority of chick provisioning. Therefore, the telomere dynamics of females may be more responsive to factors such as nest helpers and food availability because these alleviate the costs of reproduction. Alternatively, it could be that telomere lengthening is associated with the individual quality of females, since we would expect the high quality females to reside in the better quality territories and also to recruit more nest helpers. We also found that individuals with lengthened telomeres had higher survival rates, and that was true for both males and females, which is interesting because even though we weren't able to explain telomere lengthening in the males with the factors that we chose, it still had similar consequences for male fitness. And also, the effect was not solely driven by telomere shortening, since individuals with lengthened telomeres had higher survival relative to individuals with no change in telomere length. So yes, telomere lengthening was associated with higher survival, and this fits with what we already know about the system because factors such as helpers and high food availability, which are associated with telomere lengthening, are also associated with higher survival and longevity in this population. What we found is that telomere lengthening in our system is not random because it is associated with important life history factors. what we've also shown is that both telomere shortening and lengthening provide important information on the current circumstances of an individual. But because it reflects the current circumstances, it might be that it poorly reflects the past environmental stresses that the individual has been exposed to. Therefore, single measures of telomere length are likely to be a poor indicator of the biological age or future senescence of an individual. In fact, we suggest that you could liken telomere dynamics to a thermometer, in that their length goes up and down depending on the amount of stress the individual is facing at that given time. And with that, I'd like to thank you for listening. I'd like to thank my supervisory team and co-authors of this work, and all the countless field workers, database managers, lab technicians, without which this work would not be possible.